So here we can see Libby and Marcus filling up the tubes based on if they're enzyme tubes or solute tubes. Solute tubes are going to be located along the bottom row, and our enzyme tubes are going to be located along the top side. Our solute tube will contain 5.5 milliliters of water each, 0.5 milliliters of glycol, or however you say it, and 1 milliliter of peroxide. One of our enzyme tubes will contain 2 milliliters of enzyme to 5 milliliters of pH 6 solution. Our second tube will contain 2.5 milliliters of enzyme to 4.5 milliliters of pH 6 solution. Our third tube will contain 3 milliliters of enzyme to 4 milliliters of pH 6 solution. Our fourth tube will contain 3.5 milliliters of enzyme to 3.5 milliliters of pH 6 solution. Our fifth tube will contain 4 milliliters of enzyme and 3 milliliters of pH 6 solution. And last but not least, our sixth tube will contain 4.5 milliliters of enzyme to 2.5 milliliters of pH 6 solution. In this experiment, you can see that our independent variable is the enzyme amount. In each of our enzyme tubes, we have a different amount of enzyme concentration. In turn, our dependent variable would be the rate of enzymatic activity. You will see by the end of the experiment how each tube has a different amount of oxidation happening, and you could tell that by the color of it. Our control group would be the 2 milliliters of enzyme to 5 milliliters of pH 6 solution. This will give us our baseline to compare everything to. Here we are mixing our solutes and our enzyme groups in order to yield the reaction. We will collect data at 0 minutes and at 2 minutes in order to recognize the color change that happens at the appropriate times.